Okay, so I hope you guys have been enjoying your time here. Yes? Good. Okay, so I'll give these 3 lectures today, tomorrow, and the day after. And today's lecture is supposed to be very elementary. Okay, and although it's elementary, I ask you to still pay attention. I'll give you small problems. Please do work them out. And it might be elementary, but that doesn't mean that you should not ask questions. I have a tendency to speak fast, so please stop me if I'm unclear. If something, you have a question, just raise your hand and ask me. Okay, so what I'll do is today and tomorrow, I will focus on what are called 1 locus and 2 locus models. I'll explain what that means. And the last lecture, I'll focus on multi-locus genetics. So as we go along, the maths and things will become more involved. So please, write down things, ask questions, discuss, work out these problems. Okay, so let's start. So I guess you heard some of this before, yesterday, but I'll just start still from some very basic thing. I'll give you a very brief history of the subject. And as predictable, you've seen this date before, right? Yeah, this year? Why? What's important about this date, this year? Yeah, so you all know this. So Darwin published his famous book, Origin of Species by Natural Selection, by means of natural selection. So it's a big fat book, and I confess I haven't read it, but at least there are some basic points that we need to remember. Okay, so I'll just mention that to you. So there are these small points, which is that in a population, the individual differences. Individual That's very important because the operative word here is that there is a variation in the population. And that's this word which will keep again and again coming in all these lectures. Variability, variation, genetic variation, phenotypic variation. Variation is really the key word here. That's important. So there are differences, there's variation, and some of this variation is heritable, it's inherited, only partially though. So variation is inherited, it's heritable. That's another important thing. Okay, so there are these parents which will have offspring. The parents are also different, there's some variation between them, and they pass on this variation only partially to the offspring, and some of these offspring, they may be selected in the sense that they might have a higher survival probability, or the offspring number, etc., might be larger than the ones which are not selected. Okay, so the ones which are better suited to a particular environment, better variants in an environment. Are selected. So what selection means, how do we define, etc. As I go along, I'll explain this bit. Okay, so these are the sort of simple, these 3 things that you need to keep in mind. There's variation, it's heritable because that's how it will proceed with time, and there is this concept of natural selection, which I think you all have some idea about. Yes, please. Yeah, so I think you already heard that there is only a partially genetic variation, only a part of it is inherited, right?
Right.
Yeah.
So I mean, then here it could be the single nucleotide variants, and
we'll come to that. Let's learn to walk. We'll run, hopefully. Yeah. Each generation there is variation.
So in a population, individual difference exists, right? So I take one of the populations and compared to the same population, they are different. Now, when you say variation is heritable, it has an offspring, and that offspring is different from whom?
From the parents and also amongst with other offspring in that generation.
Right. Okay.
So I'll explain this point in a second. I'll explain that. So this is the concept of what's the mechanism of heredity. I'll come to that. Okay? Yeah. So there are several more major questions. So this was in itself a very important thing to understand how evolution works. I mean, there is some understanding of that, but there were several major questions. So let me just list some of them. What's the mode of evolution? How does evolution proceed? Is it a gradual process, as Darwin thought? Or does it happen in jumps, as some others argued? Okay, so there is some understanding of that now. Well, it depends on certain parameters. Both modes work, but it depends on certain parameters. So, but let me just leave it at that. What about tempo of evolution? Is it a fast process? Is it a slow process? Again, Darwin thought, oh, well, it's an infinitesimally slow process. But what does that mean? So Lord Kelvin had this argument saying that, you know, the age of Earth is finite. It's just 4 billion years or so, right? So there must be a finite speed because of this huge amount of variation that we see, right? So what's the speed? It has to have a finite speed. So this was Darwin's point of view. In the coming weeks, you will hear that adaptation can be a very fast process. And this has been documented in many, many studies now. Several examples exist. In moths, microbes, of course. Not just these microbial ones, but large organisms like moths, finches, fish, lots of such examples. Okay, so adaptation could be a very fast process, very fast in the sense of just a few, tens of generations. Yes?
What is the difference between mode and the tempo?
Mode is, like, I give you an example, fast or slow, how is it happening? Gradually or slowly? Is it going when through intermediates which are better? So what is it passing through, for example?
Isn't that related to the tempo of?
It's related, yes. Tempo is about time. And mode here, I have a certain space in mind here, the genetic space.
Okay.
How is it working there?
Okay.
I haven't introduced that, so.
Okay.
Yeah? Okay. So some of this you will hear in the coming, yes? Not best, no. No. Adaptation means, fitness has generally increased, but it doesn't have to increase monotonically. So that would be a question mode of evolution. So does it always pass through? So like Darwin's point of view was that the population is going, from certain intermediates, each of which is better than the previous one. But that's not necessarily true. For example, Sewell Wright had a different argument, which says it can go through low fitness valleys as well. Right? So it's complicated. Okay, so let's just keep it right now here. And I think you'll hear more about these two questions in the coming weeks. I want to focus on one simpler thing and a basic thing, which is about the mechanism of heredity. Okay, so this was Darwin's assumption, and somebody was asking here. So he assumed that new variation is created in every generation. So new variation is created in every generation so that natural selection can act on it. That's very important, right? If you're all identical, what is there to select? right? So variation is a very, very important, very central concept here, okay? So, but he assumed new variation is created in every generation, 1 second, so that natural selection can act on it. But then if there is so much variation, that's too much, right? If my daughter is very different from me, I mean, she's probably not a human. I mean, it will be all very, very different. The concept of species itself runs into problem, right? So, yeah, so to keep that variability in check, he proposed that the inheritance itself has a blending nature. I'll come to this to you in a second. He proposed the inheritance mechanism is blending. So, you know, we didn't know anything about genes, etc., right? Okay. So the idea of blending inheritance is like, you know, you take milk and coffee, blend. That's sort of, you know, usual word. So the way he's thinking is that genetic material, there is some genetic material which is being passed on. But it's like a liquid. It's like a paint pots of paints, which you just sort of mix. So there's a dad's, which will give red flowers, and the mom, which will give white flowers. And the offspring is somehow takes a bit of red and takes a bit of white, so that variation is created, but it's not very different from the two parents. That's called a blending inheritance, right? Okay. So that's the meaning of blending inheritance. Okay. So that was Darwin. So this was accepted. People accepted that blending inheritance is okay as mechanism for heredity, but natural selection is not. That's kind of strange, right? Okay, so I'll just read out to you a passage, and this is a sort of famous passage by this guy called Fleming Jenkins. He was an engineer, and people used to do all kinds of things at that time. So he gave this famous argument in 1867, and let me read it to you. So he is arguing that blending inheritance, that's how heredity works, but natural selection is not the driver of evolution. So he's illustrating this by this little example. Suppose a white man to have been wrecked on an island inhabited by Negroes. Our shipwrecked hero would probably become king. He would kill a great many blacks in the struggle for existence. He would have a great many wives and children, while many of his subjects would live and die as bachelors. Okay, so there is a rare, big, variant which is very better than the rest of the population has been introduced. Okay, so in the first generation, there will be some dozens of intelligent young mulattoes, much superior in average intelligence to the Negroes. So, 1867, so we don't take words literally, but the point here being that these mulattoes, because it's a black one and a white one, and it's a blend, so the intelligence of the white man is very high, of the Negro population is low, so somewhere in between the mulatto has some average intelligence. So that's what is meant by blending inheritance. Okay? So there are these, average the better, more intelligent than the wild type population. There will be such variants will be produced. We might expect the throne for some generations to be occupied by a more or less yellow king, but can anyone believe that the whole island will gradually acquire a white, even a yellow population? So what's going on? So you have this mulatto king. He will also, because most of the population is Negro, he'll again mate. Again, the offspring would have, according to blending inheritance, lesser intelligence, and so on. So the king is no longer this superior, very intelligent white person. Right? So Jenkins' argument is, if you say natural selection is the driver of evolution, it doesn't seem to be the case. Because you see, here is a case in which a variety was introduced, right? I said variation, with far greater advantages than any sport ever heard of, advantages tending to its preservation, and yet powerless to perpetuate the new variety. So what is blending inheritance, which we buy? But what is one not buying is the fact that it's natural selection, which is tending to improve the quality or leading to the perpetuation of the better variety, right? So that was the argument you could do to Jenkin. Okay, but as you all know, it turns out it's not a natural selection which is the problem. It's the blending inheritance as the mechanism of heredity which is wrong. So that's what we realized later. This is fine. What is not fine is blending inheritance. And that's due to Mendel's experiments. Because natural selection means that you choose the better one, right? The better one is selected. Right. So the white king is being selected. Mulatto is the one which is selected. That's a better variant. It will have more offspring, etc. But what's going on with time? Everybody would become the same intelligence. So that better variety is not able to overtake the population. That's what you expect. It's selected. Oh yeah, good one. Yeah, you expect that that's the one which will take over the population. But that's not what is happening here. This example. Yeah, so that whole population should become as intelligent as the invader, right? But that's not what is happening, because the culprit is not natural selection, it's the blending inheritance, because of which the average intelligence is decreasing. You know, that's the point here. Right. So I think the argument was really for the rare variants. But if you have lots of variants, then it's not such a big issue. So I think that was discussed too. Yeah. So the question is really about this limiting case where the rare variant arises. There what happens? And that's a natural situation. You know, we all have some certain genes, etc. A certain rare variant arises. Would it spread? Right? Okay, so that's the sort of question that we keep asking a lot in population genetics, and I'll try to come to that, formalize that question in a little while. Okay, so let me just come quickly to this one, 1865, Mendel's experiments. So this, all of you know, right? Okay, so I'll just go through this quickly. So I'll just summarize. So we had done this experiment with pea plants, and he looked at about 7 traits. Let me say, take the example of flower color. White or purple. So what he did was, in the experiment, he first established the pure lines. There's a patch which has white flowers, there's another patch which has only purple flowers. So then you take any 2 flowers from the white patch and make them or sell them, you always get white flowers. That's called the pure line. That's always the first step when you try to do such experiments. So, okay, and then he crossed them, so the white and the purple, and the experimental result was that he got purple flowers. This, so 2 for the next generation, and then he selfed them. And then, as you know, he got both white and purple flowers in the ratio 1:3. Okay, so this was a sort of experimental. This was a sort of experiment, and now how do we understand that? So this is something you know. Similar experiments were actually carried out by Darwin as well. He also found this 1:3 ratio. Not with flower color, with stalk length or something like that. He did. Now, Mendel's work is very important because he did not just say, it's 1:3, finished. He came up with a model to understand that. So that's really the very important significance of this Mendel's work. So he hypothesized that the inheritance is not blending. It's not like some liquid, fluid, which is mixing. It's a particle. The model, I'll use the modern jargon to say what this model is so that I can introduce to you what these terms mean. So he denoted, so we have heard this word phenotype, which in this case, the word pheno simply means something that you can see. So the color of the flower is a phenotype. My height, your height, is a phenotype. The color of the eye, etc. These are all examples of phenotype, something that you can see. Right? So the color of the flower. So he denoted this color of the flowers by a genetic material called genotype that I'll denote as A1 or A2. So what do I mean by that? So what one is thinking is the following. So now we know there is a long DNA sequence, etc., and there is some part of that genetic sequence which codes or which is responsible for the color of the flower. So I'm not going to worry about the rest of it. Just focus on that little part. That little part or the location which is coding for the color is called locus. Locus meaning location, right? And that whole bit, I'm not going to look at right now the details of which nucleotides are there. The whole bit I'm going to denote by a single letter A. If it is A1, it means this part is coming from mom, let's say, and this from dad. Okay, so that's why I write this slash. So just to say, there's two copies of chromosomes. One is from mom and the other one is from dad. And by A1, okay, so these genotypes are related to phenotype in the following manner. If I have A1, I'll call that a white flower. It's a purple. There are two colors. So corresponding to the A1 means, okay, I should just say that. So on that locus, I have two alleles. So there is protein sequence. This part is called locus. It can have two variants, A1 or A2. So if this is mom, I'll write A1 here. That's what I mean by, let's say, this guy, A1. Are these terms okay? People are okay with that? What is phenotype? And that's what I mean by genotype. It's just A1/A1, A2. Yeah?
Is it possible to know exactly what the sequence is responsible for the color?
So for this, yes, we know now. We do know it now. Yes. But let's stay in 1867, 1865, where of course he didn't know. But yet, so I think that's the idea about such schools. Like this is a school about quantitative systems biology, right? Quantitative. So you get some number like 1:3, but model it so that you understand what's going on underneath. Yeah. Yeah, that's the question of population genetics. We'll come to it. Let's stay 100 years, more than 100 years, we'll come to that. Okay? Okay, so this is my notation, etc. And of course, I can also have A1, A2. Okay? So this is the model saying that the white flower is denoted by A1/A1 and purple by A2/A2. What about this guy? That you can understand because you see, let's see this. Because when we have cross lines, let's do the pure lines first. What would this give? If mom is A1, dad is also A1, what would I get?
A1.
All A1. That is white flowers. Right? That's what he got. Right? When he crossed them. Cross lines. What would this give us? one part, take this from mom, this from dad. Mom, dad, mom, dad, mom, dad. What would I get? A1, A2, all of them? So that tells me, aha, I got by crossing purple flowers. So A1, A2 must represent purple flower. Okay? Okay, and then we do selfing. So, what would I get? A1, A1. A1, A1, which is, what kind of flowers are there? White. A1, A2, A2, A1, A2, A2, all of them are purple. Which explains my 3:1 ratio. Is this okay with everyone? Yeah, so this is all elementary. I know you have seen this in school and college. Okay, okay. So this was a sort of a basic setup, in 1860, mid-60s or so. Okay, but unfortunately, Mendel's work got lost for whatever reasons, and then in 1900 it was rediscovered by Hugo de Vries and many other people. And de Vries, he coined this term pangenes. So from where we use this word genes nowadays. Genes from Genesis, like in Bible, or just origin, that is. Okay. Yeah, we will not really distinguish. They're the same. They are different, but they code for the same color. Yeah. Three possibilities. Yeah. Yeah. Yes. Yeah, and that's what is there at the genotype level also. So this is a very simple case in which just one locus, so this is the locus, now you understand what is meant by locus, right? That locus was coding for the color. But that's not what happens, for example, things like complex trait or quantitative traits, like our height. That's very complicated. And I'll come to that in the third lecture. Okay, so right now, let's do simple things and stick with one locus problem. So that phenotype and genotype are basically same things. Okay? Okay, so fine. So this was all, and in 1900s, people started asking more questions because we have Mendel's laws. And then, what happens if I have some forces of, okay, so let me say that again. Okay, let me just rewrite this. Okay, so let me go back. Early 1900s. So this is the time in which this subject of population genetics was started. So I've skipped a lot of details here. If this history interests you, there's a nice book by this guy called Provine that you could look up. Okay, so what is this population genetics? What is this? I mean, I'm sure we don't read about that in school, right? So I'll just say that in the way a physicist would approach the problem. So just like in physics, we know what are the fundamental forces. We have certain forces, and we put them in our Hamiltonians, etc., and then we investigate what happens. Some basic evolutionary processes also have been identified over the span of all the 100 years or so. So we know basic evolutionary processes. Okay, what are they? One is natural selection, which says better one is selected with some maybe probability, or it has a better chance of being propagated, etc. So you have some idea about what that is. Okay. Then there is mutation. Again, you have some idea about what that is. That is just changing. If there's a mutation occurs, one genotype could be different from the other one, right? Now here is a new one. It's called random genetic drift. The idea here is this. So somehow it's not appreciated very widely, but much of evolution is actually stochastic in nature. Okay, so that stochastic nature is encoded in this long title called random genetic drift, and I'll consider two types of models. One that are deterministic, which means this is absent. And I'll also consider stochastic models. Okay? Why consider deterministic models? Because they are easy to understand, relatively easy, and also that gives you intuition so that you can go ahead and tackle the more challenging problem of stochastic models. So I hope to illustrate that as we go along. Yep. So this drift is not the same as what you use in stochastic processes, first of all. So this is encoded in the size of the population. It's related to the variance in the relative variance. Width. Yeah. Position in the frequency space. Okay.
Sorry.
So we'll see that.
Question.
Yep.
According to what you are saying here, stochastic, deterministic drifts, is it possible to have a purple flower when parents are white?
Yes, I can throw a dice, and sometimes. So when I say 3:1, it's not exactly 1:3. It's not like he got exactly 200 and 600. There's some fluctuation, right? But you can neglect random genetic drift if the population size is pretty large. Here, I don't know, some 10,000, some few thousands of flowers he looked at, which is pretty large. So you can neglect it, but in many cases you can't. And then you have to see. And it also gives rise to qualitatively different effects. So it depends upon this factor called population size.
Can you get a purple box?
No, unless there's a mutation. Not just due to drift, you won't. No.
Was that the question?
They will not produce purple flower, but what I might wind up having is that I have a few white flowers, few purple flowers, and I might find that, oh, somehow I got the whole population to be purple. Not because the white gave rise to purple, but because the purple flowers maybe got more sunlight. They got sampled differently from the white ones, and not because white transformed into purple. That would happen because of mutation. So I'm saying if I have population of whites and purple, it may happen that all became purple. All are purple, not became. They will become so only by mutation, not because of stochasticity. If they are two whites, they will give whites. But if you look at the population now, I have 10 white flowers and 900 purple ones. Then after some time, it might happen that the whole population is of purple flowers. It happens simply because maybe they got less food, the white ones, perhaps. Is that what you mean? Oh, no, so that's here. That's here. That's purple. So the A2 is dominant. Is that what you meant?
Maybe the question
I think he's thinking with this drift. No.
about even this experiment. Maybe worthwhile just why when you have A1, A2,
Hmm.
it shows up as purple and not as white.
Yeah, so it's just that that's called a dominant allele. So there is a fitness which is higher of A2 than A1. Okay, I hope I'm not confusing you more, but let's go ahead and see. Okay, so these are the three things that I said. And then there is a sort of broad term called population structure. So that has lots of factors in it, like age structure. So if in a population of the old ones and young ones, etc., ploidy, geographical structure, islands, two islands with a river in between, etc. So all that is in the last category called population structure. So I will not worry too much about that in these lectures, and so on. Okay, these are the sort of four processes or four forces, and what we are trying to do is to ask what happens if due to the interplay of these forces. So just like in physics, what you will do is perhaps turn off some of them, turn on some of them, and track certain quantities and see how they change with time. The quantity of interest here is what is called allele frequency or genotypic frequency. Allele frequency, which is that's the frequency of A1 or frequency of A2, or sometimes genotypic frequency. That is the frequency of these guys. So that's what population genetics is about. You track these frequencies as a function of time, or ask what happens due to interplay of these forces at large times, okay, under the action of these forces. Yeah. Yes, so the criteria was the size of the population. The population size is very large, and I haven't demonstrated that to you. If the size of the population is very large, the fluctuations, it turns out, are negligible. Okay, so I'm doing an experiment with this 10,000 plants. Probably those things are unimportant, the stochastic effects. Okay, but if there's a small population, as this white man landing on an island, here I expect stochasticity is going to be very important.
One is adding to explain certain phenomena that is strange.
No, it's just encoding a physical parameter that is the size of the population. Right? So the population size. So the physical parameter here is size of the population. Okay. Okay. So that was a sort of very quick overview of what I want to say about this little history. And then I want to go ahead with these simple one locus models. And now I want to write down some simple equations. And then let's see what we can learn. Is this much okay with everyone? Are there any questions? Okay. So let's now start to write down some simple stuff. So there was a little calculation that was done with these two guys, Weinberg. He was a physician, German physician, and Hardy, as you all know, was a mathematician. So they did this calculation at the same time, but somehow Hardy got more credit, Weinberg's work got lost or not understood. But later on, people have now, the calculation that I'm going to do is called the Hardy-Weinberg equilibrium. So let me explain what I mean. So let's say the questions. The question is this. So I give you this example. The blending inheritance is depleting variation, right? Because we said that, you know, average intelligence, the color, etc., was getting homogenized, right? That was not able to perpetuate the good variety. So blending inheritance was depleting variation, and we need variation for natural selection to act. The question is, if I buy Mendel's inheritance laws, then what happens to variation? That is the question. Is it preserved? Does it increase? Or does it keep decreasing? If it keeps decreasing, then we're still in a problem. What this calculation will demonstrate to you how to write down these equations, and the lesson that we'll learn is that Mendelian inheritance is the right way to go. So let's go slowly and write down this model. Okay. So let me write this table. I have these 4 genotypes, the one that I introduced already. Okay. And there is mom and dad, so there's a female frequency and the male frequency. And I said in population genetics, I'm interested in understanding how these frequencies change with time. So that's my quantity of interest. I say they are equal number. And of course, x plus 2y plus z is 1. Okay. So I have this population of males and females, A1, A1 type, A2, A2 type, and A1, A2 type, and they're in equal numbers. Initially, the frequencies are x, 2y, and z. Okay. And if there are no evolutionary forces and I just use Mendelian inheritance laws, how do these frequencies change with time? So let's write it down. So what is the frequency of, by prime I mean in the next generation? Maybe I'll draw this table and then we want to look at the matings that will give an offspring of type A1A1. So let's say if mom is A1A1 and dad is also A1A1, would it get an offspring of A1A1? Obviously, right? All matings would give rise to that. So probability of that is 1. Which other matings would give rise to A1A1? Yeah? Of these genotypes. Yeah. In the male population, the female population. Which other matings do you think can give me A1A1? A1A1 and A1A2? Right? That can give rise to this offspring. What's the chance of that? How many such matings will be successful? Mom, dad, right? But this also will work, this too will work, right? So chances are very good. So similarly, right? Same object I wrote. How about this one? Would it give rise to A1A1? 1 by 2 or 1 by 4? A1A1, A1A2, A2A1, 1 by 4. Okay. Right. So this is this little bookkeeping I have to do. Okay, and then I can write down the question to this answer. Answer to this question, sorry. So, x prime is therefore equal to x into x, right? 1. That's a successful mating. Then what else? X into 2y. Probability is half. There are 2 such cases. All right. The last term is 2y into 2y. Everyone okay with this? Yeah, that's simple. Okay. So you have to write it down carefully, slowly, and you'll get it. Okay? So, yeah. I'm saying that if I give you today, I measure the frequency, if it's XYZ, if there are no evolutionary forces, can you predict what would it be in the next generation? And I'm saying it will be X plus Y square, right? Okay with everyone? Math? Okay. So I won't do the next ones. I'll give you as an exercise. Repeat this kind of stuff, and you should be able to show that 2y prime similarly the same stuff you have to do, just a little bit bookkeeping, it'll work out. Please do try. Okay. So that's the frequency, yes? This one? This guy? Yeah, so there's an interpretation of that, which is that the frequency of this, so you could sort of, you need to do a two-line math, but that frequency is basically the frequency of this guy, of the A1 allele. Okay, so let me just write it down. So exercise again. The frequency of genotype A1 is actually equal to frequency of allele A1. That's the meaning of this, but you need to do two-line math for this. I'm leaving it as an exercise. Try it. If you don't get it, please feel free to ask. Okay, that's the interpretation of that line. Okay? Okay. So what? So we did one generation. Let's go one more. That's not difficult. So x double prime would be this, right? Because, yes, the next generation. So x and y is today, and prime is next generation, t plus 1. Okay, so that was also using this, turns out to be x prime. So the frequency at t plus 2 is the same as the frequency at time t plus 1. And similarly for the others. So what? So it's saying frequency is not changing with time after just one generation. Right? I have reached in just one generation, in the absence of any evolutionary forces, to an equilibrium. When I say equilibrium or steady state, it means the frequency is not changing. So x double prime was same as x prime. It's not changing anymore. Right? That's a mathematical thing. Okay? And this, there's a relationship between these frequencies that y square equals xz. Therefore, this equilibrium, this equation is called Hardy-Weinberg equilibrium. This relationship. Why is this useful or important? So you will have to use normalization. You'll get it. Yeah, but x plus y plus z, x plus 2 pi y plus z is 1. Use normalization. Yeah. 2y double prime is 2. The normalization will work. Yeah? Okay, so why is this important? So it's saying I started with a frequency x, y, z. It changed to x prime, y prime, z prime. So I still have some variability in the population in the t plus 1 generation. But in t plus 2, it's the same. So variability is not getting depleted anymore. it's preserved. So if I started with the frequencies such that they obey this relationship, they will not change ever if all evolutionary forces are turned off. Okay? So all that I have used here is the Mendelian inheritance laws. So the key point that this little exercise makes is the Mendelian inheritance is the right one. It's actually, at least it's useful because it's not depleting the variation as blending inheritance was doing. That's the key point of this little calculation. Is this okay? Yes, in just one generation. For one generation, they'll change. And if, of course, if you start like this, then they will not change at all, ever. That's called equilibrium, right? Okay. Yeah? Okay, so I'll just give you one little exercise. Please work it out. So, I just argued it's good to show things. So I'm asking you to show that if you assume blending inheritance, variation would deplete exponentially fast. So let me explain the problem. So this is taken from Fisher's book, chapter 1. Okay, so the question is this. So I have a population, very large population, because I don't want random genetic drift. I have a population, and let's say the average height, that population is A. Okay, so, and I have a couple, okay, a mom and dad. Let's say I denote the height of mom. So that's A plus some deviation to it, because it's of an individual. And similarly of dad is A plus delta D, because the average is A, average over the whole population, average delta M, that must be 0, right? So this is in the parent generation. And the variation, by the way, there's the variance here. You can check. Is simply this, assuming that mom and dad contribute equally. Okay, so this is what is given to you. I'm asking you if in the offspring height would be the average of the mom and dad height. Okay. Find the variance in the offspring generation. When I say blending inheritance, that's what I mean. You just take the heights, my mom's height and my dad's height, and I just take an average. You need not take it half, you could take some other factor, but let's just take half right now. Okay, that's what I mean by blending inheritance. Instead of Mendelian inheritance, if you assume this, what happens to the variance in the population? Please work it out. Okay, so I leave this, okay? And now I want to turn on some forces. Of the parent generation. Taking average over the entire population in the parent generation, that would be the variability. Okay, I'm asking you if now offspring will have A plus delta M and dad, then what's the variance? Okay. I didn't catch it. So I'm saying the average height of mom is A plus delta M. This is this. Mom's height and dad's height divided by 2. This is the average height in the population. Yeah, and this is the average height of an individual, which is A plus something. So the average height of all of us is something, plus, your height, my height, that's my delta M, delta D, etc. No, each of them should be 0 because the average height in the population is A. This is a random variable, its average is 0. Because if I average over all the moms, all the dads, the average is supposed to be A. That's why.
It's allowing for the fact that moms might have smaller height on average than dads.
Yeah. No, so this is within the population, within the subpopulation, I'm saying.
You have A and B, and that would make a difference.
Yeah, I think, yeah. That should, yeah, I'll just call it A plus B by 2 or something like that. A plus B by 2 if I say equal number. Yeah, that's fine. Yeah, so I'm saying mom and dad, this is egalitarian. Yeah, I'm as tall as you. Okay. So this was the simplest thing. Looks a little tedious, but if you work it out, you will say, aha, why did I spend so much time? Right? But please do work it out. Okay, so now I want to turn on the forces, and in particular, I'll turn on selection. Yeah, so actually this is useful because we assume that we are going to count individuals at the time of conception. So if you do that, there are no evolutionary forces present as a hypothesis. Okay, at that time, they're supposed to follow Weinberg equilibrium at the time of conception. So that time selection hasn't. Sorry? So I'm saying I have selection and whatnot, everything is there. But when there's a conception happens, at that time there's no genetic drift, there's no selection, it's just random mating. That's all there is. That's what is assumed at the conception, at the time of conception. So there, you're supposed to see this Hardy-Weinberg equilibrium, and people do measure it. So the deviations are seen from Hardy-Weinberg equilibrium because I'm assuming random mating, meaning this A1 can mate with that A1 A2. It's a large pool, but there are deviations when you don't assume that. There are two separate sexes, for example, they don't equally contribute, then there are deviations. Yeah, experimentally people do check it actually. They check it so to make sure that the random mating assumption is okay. Yeah.
So you can construct from the laboratory situations where the Hardy-Weinberg equilibrium is realized.
Yeah. That is done. That is actually just to check random mating. That's all that is.
What kind of an example, laboratory example is that?
I'll have to check it. I can give you a review. I mean, there's a nice review. I can give you that.
But in real population, always find departures.
I mean, random mating is the assumption. And it's okay if, and it's quite a broad thing in the sense that certain things I can change, it still holds, but certain other things it doesn't hold. And there's a theory for that. Okay? But I mean, theoretically, it's useful because of this guy. It helps me to work with allele frequencies instead of genotypic frequencies. I don't have to keep track of mom and dad and everything. I just focus on A1 instead of A1/A1. So there's a theoretical importance of this. And experimentally also it's sort of seen. It's checked. Yeah, right. Yeah. I would view it like that, yes. Departure from equilibrium? No, so I'm saying equilibrium is reached, but the frequencies might not obey y square equals x set. So, yeah, so the frequencies are obeying a somewhat different, they still reach equilibrium, but the condition is not y square equals x set. The meaning of this is just this, that frequency of A1, A2 equals frequency of A1 times frequency of A2. But you need to add and subtract some things to see that. That's the meaning of this little condition. Okay? Yeah.
Blending, I mean the term itself, right?
Yeah, actually we want to throw it out, right?
No.
No, let's throw it out,
Okay.
right? Because it's not useful. So I'm just giving you as an exercise to see what it means by blending. It's not useful.
Okay.
Okay, so let's put on selection. So this is again a very easy calculation, but it was useful at that point. Okay, so I'm going to simplify things further because my motivation here is not so much to explain you everything. I just want to introduce to you some concepts. So in the previous examples, I assumed the population was diploid. That means there were 2 copies, mom and dad. Now onwards, I'll assume that there is only one type. These are asexuals and haploids. It's just, you can do the same calculations, but it's just a little bit more involved. But you can always look up a book for that. So I'll assume haploid asexual population for now. Okay, because I just want to investigate what selection does. So I'll have just these 2 types, A1 or A2, not that A1/A1 type of stuff. So these are my genotypes. Again, we'll track the frequency. And for selection, I need to introduce to you what is called fitness. So x1, frequency of A1, and of A2 is x2, which of course is equal to 1 minus x1. And these are the fitness. So fitness is simply a measure of, you can measure it in laboratory. So for example, if you have a bacterial colony, you take a sort of, okay, so I want to measure the fitness of a virus, for example. I just make a lawn of the bacteria, drop a virus particle, it'll eat it up, it's a bacteriophage, and then you just measure what's the area that it is eaten up. So that's a measure of fitness, for example. There are many, many ways and many other things that can be used to estimate fitness. Okay, so like a growth rate or like resistance to antibiotics, etc. So I'll not worry about that right now. I'm just going to give some values. The frequency of this, the fitness of this one, this type being 1, and that of is 1 minus S, S being positive. Okay, so I won't write. 1, A1 is 1, of A2 is 1 minus S. These are the frequencies. Just like that we wrote X, Y, and Z, analog of that. Fitness, yes, you don't like it? I'll call it F1. Better? Yeah, so that's good. Okay, it's some numbers, positive numbers, let's say. Okay? Okay, so good. So what do you expect? If there's some fitness, there's nothing else, a selection. You expect that the better one would win out, right? So, and I said, think of fitness as a growth rate. So I would expect x1 prime to be like f1 x1. Why? Because that means x1t is going like f1 to the power t, right? Similarly, right? But that means this population, both of them are just exploding. They are going to grow exponentially. And I want to keep the population size to be fixed. I want to still keep x1 plus x2 to be 1. So I need to add a little extra term to this. And I'll write, to take up the normalization constant, normalization account. I'll write it slightly better as similarly x2 prime. Okay with everyone? Because you see, when I add this up, I get 1, right? Denominator is simply a normalization constant. Okay? Okay, so now coming to what you were saying, why you wrote 1. I did that because you see, let's look at x1 prime. I can write this as it's a function of relative fitness. So when I say I'm measuring fitness, I don't mean absolute fitness values. We are always measuring relative fitnesses. That means I take one of the mutant and assign it some number, whatever you like. And relative to that, somebody is fitter. It's not fit, it's fitter relative to somebody or worse off. Okay? So that's why I chose that F1 to be 1. Okay, so that's one thing. The relative fitnesses matter. And of course, this equation is nonlinear. It's a simple equation, but it's nonlinear, right? Because x2 is 1 minus x1. Okay, so we can ask the same kind of question about what's the equilibrium here, just like we did for Hardy-Weinberg. That is when x1 prime equals x1. That is stops changing in time, right? And this you can work out. Okay, so yeah, so the reason I said nonlinear equation, when I have nonlinear equations, do I have a unique equilibria? There can be many equilibria, right? Okay, so in this case, if you plug in all this stuff, it turns out, what will happen at large times? What do you think? So x1 is 1, x2 is 1 minus s, and their frequencies are going to change according to this equation, right? What do you think will happen at very large times? What's your intuition? Proportional to f1, yeah, yeah. At very large time, what will happen? It's growing at the rate of f1. Yeah, no, but what will happen at very large times? Frequency will stop changing when? Equilibrium means frequency is not changing anymore with time. When would that happen? When x1 star is half 1. Yeah, very good. 1. It's a better one, right? You expect it to win the game, right? And of course, when x1 has become 1, x2 remains at 0. There's no selection. Everybody is of the x1 type, the A1 type. There's no selection, right? Not only that, there's one more equilibrium, 0, right? Okay, so to the non-biologist, I will leave this exercise. You should worry about which, there are 2 equilibria, which one of them is stable. So usual linear stability analysis if you do, local, okay, show that x1 star equal to 1 is a stable equilibrium. Okay, fine. Okay, so this was about equilibria at large times, but now let's me worry about dynamics. That means how these things are changing in time, how this frequency is changing with time.
One little question to be clear. So, relative fitness, what is it and how is it different from this fitness that you first defined?
No, initially I said this. These are absolute numbers, F1 and F2.
And later you said that you're never fit, but you're fitter than somebody. So what that's F1 meaning there by itself?
I just mean that this is I1 minus S. If you don't like F1, you can call 2, and then F2 would be 2 times 1 minus S.
No, I mean, you can call it whatever you like, yes, but then it's 1 against who or what, you know? I mean, the concept is relative to some other population.
Yeah, so I chose to have a relative fitness with respect to fitness of A1. So usually you do it with respect to wild type, the common variety. Relative to that, you measure the fitness of a mutant in more general cases. Here I had just 2 types. If I had lots of types, I just the common variety. And with respect to that, I will measure the fitnesses of the mutants. So what one does is actually this kind of competition experiments in which you have the common garden variety. You put that with a mutant and see which one is winning. So relative to that, you will say it has won the race or lost the race, the mutant. So here I am interpreting that as the average number of offspring. The way I have written it, this is the average number of offspring produced per generation. That's my interpretation, but you could come up with other possibilities as well, right? Okay, yeah, so let me just finish this bit about dynamics. So we have this equation, and basically you could iterate it, right? But it's a nonlinear equation. You could iterate it, I mean, it's not a big deal, okay? So you could then iterate this equation for x1. You could do that, right? But I want to introduce to you a simpler version of these models. This is called a discrete time equations. They're typically harder to handle. Let me introduce to you what are called continuous time models, which are much easier to write down. So the idea is this. Suppose the fitness advantage or disadvantage, let's say of S, this S was small, then what would happen? So I'm starting with a certain fraction of A2 and certain fraction of A1, and the fitness difference is tiny. So what do you think? These allele frequencies of the A1, let's say, it would increase with time, right? Because you said that it would become 1. So let's say I started with 1/4 of A1s, right? It will tend towards 1, it will increase. But if the advantage or disadvantage is tiny, it would change continuously, right? So what I'm getting at is, if S is small, which it typically is, then I can write this not as a discrete time equation, but a continuous time one. Okay, so when S is small, because things will change slowly, like a continuously, right? So this I can write dx over dt, and just work it out, and you will get this is easy to iterate, right? This is easy to integrate, and therefore I can know x1 is a function of time. What is the meaning of this equation? Let me just spend 1 minute at this. So we understood this, right? We said that's the growth rate, or rather the growth rate is log of f1, right? So in the continuous time, if I did not have this term, x1 will go like e to the power st, right? Okay. So the general equation is this. You write down the fitness of this, just like we wrote over there, but not rather a log of that. Okay. And this is the growth term, and to keep track of the death, so this is the birth term, and then same normalization. Okay, so I should call this little f. So that's what, if you plug in over there, little f1 would be 0 for the mutant, for the A1, and it will be minus S for the other one, right? So the continuous time, you work with the log of those fitnesses, and these fitnesses are called Malthusian fitnesses. This is the Wrightian fitness, and this little f1 is called Malthusian fitness. It's just easy to write down those models compared to the relatively, to solve, it's easy to write down the continuous time equations. Okay, let me just finish this bit of the story. This you can easily work out, and we'll get, this x1 is there, yes. Because if I add these two equations, this is the average fitness, right? It is the dimensions of the fitness, just like this guy. Fitness into frequency, fitness into frequency. Okay, let me finish this. These fitnesses are the growth rate per generation. There is a per unit time, yes? Is that what you mean? Yeah. So S has a dimension of 1 by T. S in the continuous time. Yeah. So this dt is the small s. That time that I'm saying that, continuous time, that dt is like s. No, the dt is s. The dt, when I make continuous time assumption, is s. dt should be s. S is small. Yeah. Yes, so this is fine, right? Then, yeah.
That's okay. This S is not the same as that.
Yeah.
There is a time scale there.
Which is, yeah, so this time is after scaling with this little dt. Is that what you mean? Okay, so everything is dimensionless. Let's not, I'm sure it's okay, but we can think about it. Okay, so let me just get on with this because I want to make a point here. Okay, so you see, if you just solve this little equation, this is what you will get. And how does this curve look? Any guesses? Yeah, it'll increase like that and go to 1, right? So I'm plotting x1t as a function of time. This is my x1 0, it reaches 1, right? So the time scale over which selection has acted to propagate the better variety is of the order of 1 by s. And that's important or interesting because you see, s is of the order of typically 10 to the power minus 3, 10 to the power minus 2 or so. So this time is finite, it's of the order of 1 by s, unlike what Darwin was saying, infinitesimally slow process. It's not a number which is of the order of the age of the earth, for example, right? So it's some finite, reasonable number. And that's what a sort of point which was made in this original calculation of Norton, Punnett, Haldane, etc. That there is a time scale which is of the order of 1 by s. Okay? Right? So that's what this little point it makes. The time scale over which the favorable mutation would take over the absence of other things is of the order of 1 by s. I'll just take 2 minutes because I want to introduce one more force, which is mutation. And it's very straightforward, much simpler. So, okay, so what did we? So the effective selection was that it homogenized the population, right? Everybody became A1, A1, A1. So the thing was that natural selection is not the one which is propagating the best one. But yes, it is doing that, right? The better one won. A1, its frequency became 1. That's what I mean by is propagating or perpetuating into variety. And it happened in a finite time, which is of the order of 1 by s. So selection has wiped out the variability. But so what happens now? I need variation because I want evolutionary process to go on. How do I get that? Variation again? I'll introduce mutations. So you can write in an analogous manner this little table. So we have the same x1 and x2 and fitness is 1. Only mutation is what I'm thinking about. And let's say x1 and x2, they can change into each other with certain probability. And let's write down the equation for that quickly. So I'll take 5 minutes. Let's say I'm doing x1 prime. No selection, only mutation. So there is a probability that 1 can become 2 with probability mu1 2, and there's also probability mu 2 to 1. So how would I get x1? So if I had an x1, it should not mutate away, right? Then only I'll get a contribution. Agree? Yeah? You can also get from x2, it should mutate to 1. Anything else? Normalization? Is this okay? Yeah? I don't need normalization here, it's normalized. I mean, I don't need any denominator here. It is a linear equation. Okay? So this is easy to work out. Equilibrium and dynamics, the same questions that I asked. So the equilibrium. If this mutation rates were equal, simply half. This is what you expect intuitively as well, right? Dynamics are also very easy. I'll just give you the answer. Okay, so here the time scale is not 1 by s, but 1 by mu or 1 by 2 mu. So over this time scale, mutation would generate variability. Mu, yeah, when they are equal. Okay, the time scale here is of the order of 1 by mu. And just one sec. So with this mu, this mutation rates are pretty small numbers. The selection coefficients, that S that I said, that's of the order of 10 to the minus 3 or larger. That's all we can detect. 1 second. And this mu is pretty small, like 10 to the minus 6, minus 11. For RNA viruses is larger. But the point here is that mutation will take a long time to generate variability. Selection is wiping out the variability. Mutation would generate, but it will take a long time. Okay, so I want to finish this bit. I'll take the question in a minute. I'll just take this one last minute. So what did I do? So we started with no evolutionary forces. I put in selection, then I put in mutation only. Obviously, there are all these forces present. So I'm leaving this as an exercise for you to think about. Suppose I have both mutation and selection. Then what will happen? So I have the same model, but now fitnesses are not 1, but they are 1 and 1 minus s, and they can change into each other with a probability mu mu. It's a straightforward generalization of what I did. Please do work it out, and I'll ask tomorrow what you find. If you work it out, you'll find something interesting, and I think Luca would say a bit more about that, because it will demonstrate to you in a very simple way a phenomenon which has been seen in certain experiments as well. Okay, so please do try. Okay, question? Yeah. It's easier that way because, yeah. Here also you can do, it's not difficult this case because it's linear. Mu is, both probabilities are equal to mu.
Let's be.
As a natural selection and?
And that also predicts long-term behavior. So how can this model work? Means where the heredity is not taken into account, only selection is taken into account, like replicator equation.
Yeah, so but those are density-dependent selection equations, right? Yeah. So, sorry, if the density is sorry.
Yeah.
No, but that's the F's are not dependent on the density here. The constants. That object is the average fitness.
Yeah.
So then if the frequency is, so what happens in those equations? If the frequency is decreasing, fitness might increase. The fitnesses are also going to change with time in response to density, right?
But in long-term picture, they predict the long-term
So
picture.
here, the best one won. But here, when you have replicator equations, there's no best one. It will keep changing, no? Fitness is.
Yeah, I'm just asking, their heredity is not taken into account. Means
Here, I have taken into account, I said haploids, but if I had done this calculation with diploids, I will again do this thing, a mom, dad, etc., the same as what I did for Weinberg equilibrium. I will do that. So everything is Mendelian inheritance. Blending inheritance, we're not even going to think about anything, except for that little exercise I give you. Anything else?
Heredity is taken into account.
Right.
Yeah, but I was asking from the point of view of replicator equation, where everything is decided only from the fitness.
Yeah, so but I'm saying fitness is going to change with the frequency. Frequency is going to change, so with the fitness. Here, fitness is constant. So I know this is the best guy, right? But there, frequency will change, fitness would change. I don't know who's the best guy. It'll keep changing with time, right? So I could have variability there. Yeah? Okay.